Neurons form their connections through axon growth, arborization, and synapse formation during a developmental process known as wiring. Identifying the mechanisms by which neurons are precisely wired in specific circuits is critical to understand how the extraordinary complexity of brain function emerges. There are two main classes of neurons in the mammalian cerebral cortex: glutamatergic pyramidal cells, which are excitatory projection neurons, and GABAergic interneurons, which form inhibitory local connections. They have different features regarding their morphology, connectivity, and firing patterns, and these differences are built by specific molecular programs. So, what are the molecules that are mediating the differences between these two populations? The molecular mechanisms regulating axon development and synapse formation in pyramidal cells have been studied in detail, but our understanding of these mechanisms in interneurons is very rudimentary. To understand how this heterogeneity emerges, we search for differentially expressed genes between these two populations during cortical wiring. We use FACS to isolate cells, and later on ran transcriptomic analysis. More specifically, we labeled pyramidal cells and interneurons using mice reporting these populations with GFP, and compared the gene expression levels of the different conditions. We identified a gene set of 133 genes specifically expressed in interneurons during wiring. One of the first candidate genes was the serine-threonine kinase Nek7. This encodes a protein involved in microtubule dynamics, which is the cycle of microtubule polymerization. In situ hybridization of Nek7 showed that it expressed mainly by a subpopulation of interneurons, the ones expressing Parvalbumin. As Nek7 has been shown to mediate microtubule dynamics, and this is essential for axon and dendrite development in neurons, we thought Nek7 could also regulate microtubule dynamics during the maturation of interneurons. To know Nek7 function, we depleted its level in interneurons and observed the effect. We did so by engineering a Cre-dependent conditional vector expressing an shRNA against Nek7, together with the fluorescent marker mCherry. We explored the dynamic behavior of microtubules as reported by EB3 using in vitro time-lapse imaging. Interestingly, we found that downregulation of Nek7 in interneurons increases the average speed of EB3 comets. To examine whether the deficit in microtubule growth led to alteration in axon development, we explored the dynamic behavior of axon in vitro. We observed that Nek7 depleted growth cones have increased turning angle, which suggests that the steering of the axon is affected by the lack of Nek7. Since interneuron axonal arbors are highly branched and convoluted, defects in axon dynamics may impact normal axonal morphology in these cells. We found that the average neuronal length of interneurons in culture, among other variables, was significantly decreased when depleting Nek7. But what happened in more physiological conditions? To investigate whether Nek7 regulates parvalbumin interneuron axon development in vivo, we downregulated Nek7 with the same vectors introduced in adeno-associated viruses, AAVs. Similar to the in vitro results, we found that the total neuronal length of Nek7 knockdown cells is reduced. There is also a strong correlation between axon development and synapse formation. Indeed, the growth of axonal arbor seems largely dictated by the formation of stable synaptic contacts. To know whether Nek7 is mediating synapse formation, we also injected AAVs. We then quantified the number of synaptic buttons made by the infected parvalbumin cells in their mere synaptic target, the soma of pyramidal cells. We found a decrease in the number of buttons in Nek7 knockdown interneurons compared with controls. So the precise function of Nek7 in axonal wiring suggests that parvalbumin interneurons use specific mechanisms for axon growth and synaptic targeting that are not present in pyramidal cells. Our results also suggest that Nek7 is part of a broader molecular program in parvalbumin interneuron wiring, but further research will be needed to investigate this.